and stuff in my office. And as a sign of my gratitude, okay,
What's ooh.
I make pins because I'm like really.
Are they nerdy pins?
Oh yeah, they are. Of course they are. So
I wouldn't expect
I expect you. There's one for everybody, including those that are not presenting. I expect you to be wearing these on Friday, right?
My backpack.
Of course. Why not? Oh, okay.
Love it. That's what I always
Are those images of ours?
Yeah, that's one of the best images that we got. So.
Do you want to take a pin?
Sabrina's not appreciative
Friday.
of your class.
Last time, right?
Sabrina's not appreciative of most anything, so it's totally cool. I think the extra one's mine. Anyway. No, it's okay. Just stab it into Sabrina. It's cool.
We're presenting during class time.
Yes, we are. So no class on Friday. So presentation side. I do. I just got it. And you and I need to talk because they effed up on the schedule. Not on just yours. So we'll talk about that later. No, it's okay. We'll talk about it. No, it's just they are assuming you're doing a poster on your research, and they didn't include your two poster on the list of posters. I'm very frustrated with it. I'm gonna leave it at that for now because I'm gonna talk about it later. All right. What, Sabrina?
Just like a general approximate.
When and where are our little
9 to 9:50 on Friday.
So during
Downstairs, yes, it takes place of class on Friday.
and so do our like little informal speeches.
Your talks on your posters? Yes, that's Friday class.
Okay. Another question.
Yes, ma'am.
Where so do you have to go to TAC to get it printed or does it print and then
So
how does that
you
work?
can there if you look at the instructions that I've made available for you on how to do this, which is with the rubric, you go to services.edgewood.edu, and instead of clicking on the button that most of us would typically click on, which is something's broken, you would click on the request something or request
Yeah, I did
a
all,
thing.
yeah, we did all that.
Did you
And I got
submit
a
and everything?
confirmation, Yeah.
Oh,
What
okay.
would I do after go to?
So they will print it at TAC, and then they will email you when it's ready. Okay.
No need
And I
to
also-
be sick.
So today we're gonna pick back up on HIV and talk about. We'll talk a little bit more about attachment, and then we're gonna move into the actual reverse transcription process and see how far we can get with that today. The goal on Wednesday then is to try and finish up as much of the life cycle as we can, and then Monday is going to be kind of a review day, but in the context of looking at antiviral drugs particularly. So I'll just kind of skip ahead to where we left off, which was right here. So we talked about with the attachment and entry step, there are a few events that take place. And there are a couple of essential proteins, both on the host and on the viral side, that contribute to this. So GP41 and GP120, which are both products of the ENV or ONV protein, or ONV gene in the HIV genome, those form basically a trimer of dimers. In other words, 3 GP41s and 3 GP120s come together to form a 6 polypeptide structure to form the protein. And GP120's first attachment is to CD4. That causes a change in the shape, revealing a second binding site, which is where which is where GP120 will bind to either CXCR4 or CCR5. And after that second binding has occurred, GP41 undergoes a change in formation, embeds this fusion peptide into the cell membrane, and then begins to kind of collapse back on itself to bring the membrane of the host and the membrane of the virus together. And again, this is a fusion process that's occurring right on the cell surface. So there's no endocytosis or uncoating in the phagolysosome in this case here. So I asked you guys to take a look at, over the weekend, anything dealing with CCR5 or Timothy Brown or the Berlin patient or anything like that. What did you guys find? Sabrina didn't do it. By the look on her face. Jonah, I'm going to hold you off because I know you know the answer. Anybody take a look at this? All right, Jonah, go ahead.
Well, there's the CCR5 delta 32 mutation, which is pretty cool.
Okay, so what is CCR5 delta 32?
It's a 32 base pair deletion from the CCR5 gene that makes that protein not really. It's like in the membrane, but it's not functional at all. It's just kind of misshapen and.
Yeah, it's non-functional, and actually it loses, that delta 32 loses the amino acids that are required for the attachment of GP120 to CCR5. So about 1% of individuals of Western European descent have this gene mutation. Individuals who are homozygous recessive, lack both copies, don't really seem to show any sort of immunological defect because there's so much redundancy in the immune system, but they actually are resistant to HIV infection. So a very small percentage of the population are resistant as far as we can tell. There are a couple of cases where we've seen this. So one individual, and I'm always mistaken on the names of everybody that are the role players in this, but there was one individual who had numerous contacts with HIV positive individuals, never became HIV positive himself, and they found that he had this particular mutation. The Berlin patient is someone who was HIV positive but also had leukemia at the same time. And they were trying to treat the leukemia, so they irradiated, got rid of all of his T cells, and then did a bone marrow transplant to transplant him with T cells from another individual. In doing so, they actually were able to, as far as we can tell, cure him of HIV as well, because the bone marrow donor had that CCR5 delta 32 mutation on both copies. So I think the last I had seen was like that individual had been 5 years without antiretroviral drugs, without any sign of HIV infection. This became very, very interesting for a number of reasons. One of the main drugs that is being used now as an inhibitor of this entry process is called Cellzentry, and it will effectively bind to. And the importance here, I think I want to point this out real quick. CCR5 is found primarily on those macrophages, and the macrophages are the first of the cells that get infected during HIV infection. At some point, there's this transition from M-tropic to T-tropic, or from macrophages to T cells. I'll be honest, I don't really understand this transition, or if there's what types of events are leading to this transition. It very well could be that HIV first comes into contact with macrophages, and as those macrophages move to the lymph nodes to present pieces of virus on the cell surface, that's where they come into contact with T cells. That's my guess, but I'll be honest, I don't totally understand the physiology of this step here. All right, So let's try to get into some reverse transcription now. The enzymes that are involved in the reverse transcription are obviously the reverse transcriptase. There are going to be other proteins that are involved in later steps of this as well. For example, integrase is the enzyme that's actually going to take that reverse transcribed cDNA and integrate it into the host genome. The three main proteins that are all products of this polymerase or Pol gene include reverse transcriptase, protease, and integrase. It's produced as a polyprotein and then is cleaved by the protease into the mature forms of those proteins. And these are all in association with that full-length genomic RNA as the virus is entering into the host. What this image is showing you here is that the polymerase itself is actually a heterodimer made up of two different forms of the gene, one known as P66, the other P55 or P51, and there are two domains. There's a polymerase domain, obviously. That's the one that's adding your A, G's, T's, and C's as it's reading the transcript. And then there's a nuclease domain as well. And we'll come back to the importance of this nuclease domain a little bit later. Now let's define what kind of polymerase this is real quick. We're starting with what and ending with what product? RNA to DNA. So this reverse transcriptase is what by definition? Good. RNA-dependent DNA polymerase. Absolutely. And these events are going to occur in the cytoplasm, likely in partially uncoated capsids. All right, so let's take a look at the steps. What I'm going to do is I'm going to try to draw on top of this to just provide a little bit more detail in terms of what directions everything are moving, and then I'll describe some of the important parts here. The genome itself contains the following, what's known as a repeat or an R region, and I'm just going to kind of note these different parts. U5 stands for unique 5', so this is a unique region of the RNA that's on the 5' end. PBS, this is going to be the polymerase binding site. And note what this is base pairing to. This is our tRNA from lysine. This is actually going to serve as the primer to begin the transcription process, or reverse transcription process. We've then got our GAG, POL, and ONV genes here. PP stands for polypurine. In other words, this is an AG-rich region. Okay, so purines refer to the base pairs adenine and, to the nucleotides adenine and guanine. Come back to the importance of that a little bit later. And then there's a unique 3' region and the repeat region again. The main elements that we're going to focus on in here are everything, primarily PBS, R, and the polypurine tract. Those will be the main focus points for reverse transcription. Now this image is not perfect. There's some flaws in the model as drawn here. They're relatively minor, but this is probably one of the best images that I've seen to describe this process. And I'll try to point out where sometimes these images differ in case you're looking at yourself at some of these processes. Okay, polymerization always occurs in what direction? If we're adding nucleotides, what direction are nucleotides being added? Perfect. So from this tRNA primer, here's the 5' end of the primer, 3' end is here. So from that PBS, at least in this image, we're going right to left. Oh, that's not what I wanted to do. There we go. So from that primer, RNA polymerase will bind, the reverse transcriptase will bind then begin polymerizing the 5' to 3' direction. It's going to reach the repeat region, and then it's going to stop. Because obviously there's no more base pairs beyond this to polymerize. So we get what we call a strong stop, and there's actually 2 strong stops in this process. At this point, it seems kind of like from what the models tell us, reverse transcriptase will pause, and then what we also find is reverse transcriptase has nuclease activity. In other words, sometimes this is referred to as RNase H more specifically. And RNase H is an enzyme that will identify RNA-DNA hybrids, which this is, and it will degrade the RNA. It'll cut apart those RNA nucleotides. And that's what this step right here is showing us. So in this step here, the RNase H activity of the reverse transcriptase chews away these RNA nucleotides. Okay. Now some models you're going to see actually show this genome here in a circular or kind of like a loose circular formation, where this R region here is looped around to this end. So since we have cDNA, the complement of R right here, this is going to base pair with the R that's located at the 3' most end of the RNA genome, and then we're going to allow for polymerization to continue. Okay, so this model is showing as if this whole thing kind of detaches or jumps from one end to the other. Really, it's more that, I'll try to draw this real quick. Nope. Really, what we would have is here's our tRNA lysine, cactus. Here's our R. And then our RNA would kind of loop around like this so that we get base pairing between those R regions. And now there's template for reverse transcription to continue. Okay. And so this would continue in that 5 to 3 direction around this whole thing until there's no more room to go. So we have that jump, continue to polymerize at this point all the way to the PBS. At this point, we've got repeats of the PBS regions. We've got a U3, R, and U5 all right next to each other. And at this point, then, the RNase H activity comes in again. And what I mean by that is all of this RNA here will be chewed up by the by the reverse transcriptase. So we're left with cDNA at this point, with the exception of one region, and that is the polypurine tract, or PP.
That's still RNA?
Yeah, that's still RNA. Polypurines are actually relatively challenging for reverse transcriptase to destroy or to hydrolyze. So this particular region is more resistant to degradation than the rest of the genome. It will get degraded, it's just that the rate at which it gets degraded is slower. So what does this serve as then? This becomes important in the next step of replication. If we look at this image down here, what does that tell us about this PP, or this polypurine tract? What's it serving as? Yeah, exactly. It becomes the primer so that the reverse transcriptase now can form double-stranded cDNA. I'm going to see how many more times I can say PP during class also, I've decided. So this becomes the primer for what we call second strand synthesis. Making that second strand, in this case it's going to be cDNA now. We'll have a point at which this becomes, at which transcription is going to stop again, and this tRNA, what's it made out of? What's its genomic material? RNA. We've now got a DNA-RNA hybrid, so what happens to this?
tRNA gets degraded.
Bingo. tRNA gets degraded, which we're seeing here, and now we get a second jump. This polymerase, or this complement to the PBS region jumps to the opposite end of the genome and completes the polymerization process. And we also get, at this point, completion of polymerization from the PBS to the end as well. Okay. So the end product of what this is, and I'll try to summarize this in a minute, or the steps in a minute, but the end process of this is we get U3R and U5 on both ends of the genome. These become what we call long terminal repeats, or LTRs. Okay. We've also got the PBS region, GAG, POL-on, and the polypurine tract as well. When mRNA is synthesized, the mRNA is going to include the following. This LTR here actually becomes the promoter, so this N becomes the promoter. And transcription will include nope, that's right. U5, PBS, GAG, POL-on, PP, U3, and R. To reproduce the original full-length genome, essentially. We'll come back to that a little bit later. So I'm going to pause real quick because that's a lot of steps. Okay? Let's try to summarize, though, what the important steps are here, or maybe not what the important steps. What are the important features? Go ahead, Sabrina.
So every time that you have like the stop or the pause, is that where the RNA sensitivity comes in?
Yeah, that's kind of how that's one way to mentally compartmentalize it, absolutely. So the polymerase is pausing, you've got RNase activity, degrading that RNA, that allows for this quote-unquote jump to occur from one end of the template to the other end to continue polymerization.
So where does it pause? Is there a specific place?
So there are a couple of places where pausing occurs. So again, if we go back to the beginning here, the first, basically it's going to pause when it runs out of sequence to polymerize. So in the first pause, it's at the R region. And then at the second pause, it becomes the PBS region. Grace.
When you say like it jumps, does that just mean like it's circular?
Yeah, the terminology that we use is it jumps from one end of the strand to the other.
Mm-hmm.
What we really think is happening is that it's not a physical movement of the DNA itself, but rather, because we assume that this is kind of like loosely circular, it allows for the polymerization to continue. But we typically in a linear form like this, we just describe it as a jump from one end to the other. So main takeaway features. Obviously, the function of reverse transcriptase. Take the RNA template, make DNA. What are other key characteristics that we see here? And maybe what I'll do is I'll allow you guys to play around with this for a little bit before we answer that question. Okay? So why don't we take like 5 minutes or so. And what I'd like you to do is, actually I'm not going to give instructions for this. Just do what works for you. Take 5 minutes and try to describe the steps, but however you do that doesn't matter to me. Or organize this information in some way that's going to be beneficial. Okay? Sabrina?
So the TNA is packaged in the various.
Yeah, we will talk later on very, very briefly. Basically, it's during the packaging step that gets included in the virion. Okay? All right. So go ahead, talk it over with somebody else if you'd like to, and then we'll try to summarize this together. I'm going to be honest real quick with this. I just want to say this before people start freaking out, like, do I need to memorize the order and everything? And the answer is no. I think that detail would be more of a take-home type question, okay? But I want you to walk away with the major themes to this. So let's try to summarize those. What are some of the major themes that we see here? Please.
The tRNA creates a primer so that the polymerase can bind to the PBS region.
Okay, that's great. tRNA as primer. What's the other primer? Good. PP.
I just wanted to say PP.
I know. Childhood humor is fun. What else? Okay, so I'm gonna elaborate on that just a bit, Paige. So RNA, I'm gonna put gRNA for genomic RNA. Serves as template for that first single-stranded DNA, and then the reverse transcriptase will take that single-stranded DNA and make double-stranded DNA from it. And if you're thinking about where do the mistakes get made in the HIV genome, this is the step. It's this reverse transcription where that low fidelity of the polymerase really can have an effect.
Question.
Go ahead, Grace.
So the reverse transcriptase converts it from RNA to single-stranded DNA.
Yes, it's doing all of those. So this is it's kind of hard. We describe the reverse transcriptase as an RNA-dependent DNA polymerase, but it's also in effect a DNA-dependent DNA polymerase as well.
So can you both?
Yeah, yeah. Other features. I could think of maybe two that I'd like you to know. One would be the nuclease activity of the reverse transcriptase. And the importance of that really is that it clears the RNA so that the second strand of DNA can be made here. And it allows for the creation of the LTRs. And I think that this is kind of the last point, because we'll come back to those LTRs a little bit. In part, they serve as binding sites for the enzyme integrase.
Can you remind me what the LTR stands for?
Long terminal repeat.
Gotcha.
And again, the LTR is made up of U3, R, U5. Those are the three genomic elements that contribute to that.
And the purpose of the LTR is
Yeah, the LTR will serve as a promoter. It also actually, I mean, if you think about it, they actually kind of almost serve a similar function to telomeres in the same way. They prevent the loss of genetic information. So you could argue that it might have that there are multiple purposes to it. But we'll talk about it just primarily as a promoter site. Go ahead.
Still in the
Yes, this would all be in the cytoplasm.
double-stranded sequence. Yeah.
Yep. Paige. That's a good question. So of the reverse transcription process, mostly what they are inhibiting is the reverse transcriptase enzyme itself. So there are two forms. There are what we call I'm trying to remember if I have the acronym right. I think it's RT. I'm not going to try to remember the acronym right now. But there are some that will bind to the reverse transcriptase and inhibit its function that do not look like the nucleosides. And then there is a group of them that look like the A's, the G's, the T's, and the C's that bind to the reverse transcriptase. We categorize them separately, but some would bind to the active site and be competitive inhibitors. The non-nucleoside ones are more like non-competitive inhibitors, but they're specifically trying to block that production of the cDNA. Yeah, that's the primary goal there. Is there anything else you wanted to explore on that right now? Okay. All right. Okay, I think we'll leave it at that for now, and then if questions come up, they come up and we'll address them later. So with what time we have remaining, I want to get a little bit into transcription and talk about, integration and then transcription, to try and address some of the questions that came up even on Friday, like the full-length mRNA and how does it decide to transcribe translate or package into the genome type questions. The integration step requires the enzyme integrase. There are integrase inhibitors. This is a category of antiviral drugs that has been explored, but there aren't many of these drugs right now. The way that we think integrase works is it binds to the LTRs. So it binds to each end of that double-stranded cDNA. Integrase actually helps the import process. So now we're going from cytoplasm into the nucleus. So to import that DNA in, integrase seems to be important for this step. And there are a couple of the other proteins, some of those accessory factors that seem to be important here as well. And integrase kind of works like both a restriction enzyme and a ligase. So if you remember, restriction enzymes identify sequences specifically and make cuts. Integrase doesn't have the specificity, but what it can do is cut double-stranded DNA, that being the host genome. It will actually chew back the sequences a little bit to leave overhangs. And then those overhangs become the sites where the HIV genome will be incorporated. And so that's what they're trying to show here then, is that integrase will ultimately ligate the HIV genome back to the host genome, and then the function of integrase is gone. And at this point, similarly to how we use the term prophage to describe an integrated bacteriophage genome, this we would refer to as a provirus. So at this point, HIV's genome is integrated into the host genome, and now transcription and replication are completely controlled by the host factors. The integration seems to be random, though there's some evidence to say there may be some areas that are more hot for integration than others. And we don't really, I'll be honest in saying that I don't know what makes a site particularly fond for integration. It might have something to do with the accessibility of that genetic region, you can imagine that heterochromatin might be more accessible than euchromatin. So areas that maybe are actively transcribed would be more likely to have this integration step occurring. All right. Transcription. So transcription with HIV can be a little bit goofy, and actually we find that there are I was reading some literature and I was trying to mentally organize this last night. There are 5 mechanisms that all contribute to the translation of genes. So some of them we've seen, and I'm just going to kind of list these out real quick before we actually talk about the initiation of transcription. So events that contribute to how often does a protein get translated. Or what proteins do get translated. Alternative splicing is one, and we saw this with HPV. So based on how mRNAs are spliced, we'll determine which of the genes is going to be translated. We're going to find polyproteins, which we've talked about in principle but haven't really seen many examples of. A classical example of this is poliovirus, where we've got one polyprotein produced and then a protease encoded by the virus itself will cut that polyprotein into mature, smaller fragments. We'll see that with HIV. Third one is something we've talked about in reading, which is the ribosome shunting. You may remember that we saw this with adenovirus, where based on like some three-dimensional structure that's not technically an iris, it allows for the ribosome to kind of jump down further in the mRNA and pick up translation. This is used in some cases where we have actually bicistronic mRNAs. For example, some of the transcripts will include VIF and the ENV or envelope proteins encoded on the same mRNA. And so ribosome shunting can contribute to which of those two is going to be expressed from that mRNA during a translation. Fourth mechanism, which is up for debate, iris. There's not been one particularly identified, but it's thought that HIV may have an iris upstream of GAG and POL. And then the last one we'll take a little bit of a look at is ribosome slippage. And this one actually contributes to a difference in expression of GAG and of POL products. So those proteins that contribute to the capsid structure, or those that contribute to the reverse transcriptase, protease, and integrase. Okay. So we'll try to come back to at least the ribosome slippage. We'll see examples of these other parts, with the exception of the iris as we're going along. So transcription starts from the LTR. And what this image here is trying to show you, cis-regulatory elements just mean these are elements within the genome itself. So the LTR actually has binding sites for host proteins to activate transcription. This part B here is kind of blown up version of the LTR. And what they're trying to show you is that you have enzymes like, or transcription factors like NF-kappa B, nuclear factor kappa B, which is very abundant in T cells and actually quite active in T cells as well. It's one of those proteins that when it's phosphorylated, kind of like ERF3 and 7, it will move to the nucleus and bind to DNA site specifically. And then there's also binding sites for another transcription factor known as SP1. Again, pretty abundant in T cells. So when these two proteins are available, we might find that there will be expression of the GAG and PAL and ONV genes and those other accessory proteins. Okay, so it's all the hosts that's driving this expression. Those will recruit the RNA polymerase, and particularly since it's mRNA, it's RNA polymerase II that is used. And RNA polymerase is going to initiate transcription. So this is not something we've necessarily talked about yet this semester, is what's the difference between the initiation and the actual elongation of transcription. I'll try to draw this out a little bit. So what we know about RNA polymerase is during the initial steps of transcription, RNA polymerase in eukaryotes is recruited to a promoter, placed right over the transcriptional start site, which is sometimes referred to as the +1 nucleotide. This is not the start codon, this is just the beginning of the 5' UTR, or the untranslated region. And it will go about 10 nucleotides or so. And if there are additional signals not present, the RNA polymerase just aborts transcription at that point, just stops. So there needs to be other events that take place that contribute to this transcription continuing. Some of those events include phosphorylation of the RNA polymerase, basically a hyperphosphorylation. And actually it shouldn't necessarily be, no, we'll leave it at that. Hyperphosphorylation right there. That hyperphosphorylation not only encourages that RNA polymerase to continue into the elongation or transcribing the rest of the mRNA, but it also removes what we call anti-elongation factors. Additional proteins that dock onto the RNA polymerase that, when they're bound to the RNA polymerase, will stop transcription at the initiation point and not allow for this transition into elongation. Right. HIV plays with this system. It ensures that from its promoter, there's not only going to be an initiation of transcription, but elongation will occur as well. And I'm going to give the brief Cliff Note versions, and we'll come back to this on Wednesday. The role players here are the viral protein TAT, and it's kind of hard to see in this image, so I'm going to try to draw it with a slightly different color. But what they're trying to show is when this transcript begins to be made, there's actually a palindromic sequence that forms a hairpin loop. TAT binds to this hairpin loop, which has been referred to as TAR, TAT transactivating region. Okay. So when TAT binds to TAR, this allows for TAT to bring, or TAT will also interact with a couple of proteins from the host. We're just going to refer to this main one, which is TEF-B. And TEF-B brings along with it CDK9 and some other proteins. Well, this leads to the phosphorylation of RNA polymerase. And when they phosphorylate RNA polymerase, that allows for the elongation to occur. Okay, we'll talk a little bit about the evidence that has led to this model. We'll probably try to start out with that on Wednesday. But with that, have a good rest of the day. To some of you, I'll see you in 10 minutes. In genetics.
Oh, you're leading genetics.
Oh, damn.